How do plants tell time? Group L, Griffin Asher, Damaya Chingrutna, Adam Irwood, Allison Welty, and C.C. Zhang. Circadian clocks in plants are influenced by environmental signals that entrain feedback loops that ultimately determine their daily behavior. This circadian rhythm in turn triggers many processes such as flowering, fragrance emission, and leaf movement. Experiments have been performed to demonstrate the effect that sugar has on circadian rhythm. They show that these sugars produced by photosynthesis regulate genes that influence this rhythm. The main gene, pseudoresponse regulator 7, is what gives the circadian oscillator feedback. Overall, plants wouldn't be able to tell time without photosynthesis and sugar metabolism.
From dawn, light activates PRR7 and drives photosynthesis. The concentrations of simple sugars produced by photosynthesis accumulate within the plant during the day. Peaking around 4 to 8 hours after dawn, represented by the red line, high endogenous sugar concentrations lead to suppression of the PRR7 promoter, contributing to the phase of PRR7 rhythms. PRR7 is a transcriptional repressor of the circadian clock component CCA1. Thus, the rhythms of the endogenous sugars derived from photosynthesis contribute to circadian entrainment through PRR7. The proposed clock mechanism represents a metabolic dawn. Dawn is a time-dependent gradient of light intensity, whereas metabolic dawn represents a gradient of increasing metabolite concentration. The metabolic gradient lags behind that of light and contributes to the setting of the circadian clock. In the model previously established, relationships are shown by black connectors, and novel relationships here are shown by orange connectors.
In one particular study from Nature magazine, cultures of seedlings were kept in one of two environments to test for the effects of the absence of photosynthesis on the genes involved in maintaining the circadian rhythm. The first environment being CO2-free, and the second being a medium containing DCMU, which blocks photosystem 2, thereby inhibiting photosynthesis. In addition, a control was prepared and tested for comparison. The results are given in the figures here. As shown, inhibiting photosynthesis in the seedlings increased the circadian period by an average of 2.5 to 2.9 hours. The reason for this increase can be attributed to the effect that initiation of photosynthesis has on a key promoter gene, PRR7. As shown in Figure A, PRR7 activity increased with the inhibition of photosynthesis, verifying that photosynthesis is in some way responsible for the shutting down of PRR7 activity. Now, what is the mechanism by which photosynthesis alters activity of these promoter genes? As shown in Figure C, inhibition of photosynthesis caused sucrose levels to accumulate much more slowly. So the study shows that sugars like sucrose may have something to do with it.
To test the general effects of sucrose on the plant, seedlings were grown in mediums containing sucrose, glucose, or fructose, and a non-sugar control. In all the external sucrose, glucose, or fructose mediums, the plant was able to maintain its circadian rhythm in a continuous dark environment. By looking at the diagram, one can see the red path, representing the sucrose, is maintaining its rhythmic cycle. This suggests that the circadian rhythms represent a general response to the metabolically active sugar. Light from photosynthesis can also have an impact on the circadian rhythm of the plant. To verify that the rhythm is caused specifically by sucrose, plants were treated with either sucrose or light. When treated with sucrose, the circadian phase is 22.8 hours. That gives it time to prepare itself for photosynthesis when the day starts. However, when the plant is treated with light only, the circadian phase is delayed and does not match the cycle that will allow the plant to photosynthesize effectively.
This experiment indicates the importance of sucrose in inhibiting PRR7 activity. When photosynthesis was inhibited, the results were similar to that of aforementioned experiments. PRR7 activity increased to high levels. Now, when photosynthesis was inhibited but sucrose was added to the seedlings, regulation of PRR7 activity was similar to that of the control. So the whole reason photosynthesis affects the circadian period is because of its effect on sugar synthesis. So in summary, when photosynthesis is not occurring at its usual pace, sucrose levels accumulate much more slowly, shutdown of PRR7 activity is delayed, and the day phase of the circadian period is initiated later in the day.
It is very clear that sugars produced from photosynthesis are essential to the regulation of plant circadian rhythm. The data from photosynthesis entertainment of the Arbedias thalianas circadian clock shows that several protein molecules play a role in the circadian clock. As the graphs shown indicate, PRR7 is induced by sugar produced during photosynthesis, as a module of the graph can be clearly associated as oscillating every 24-hour period. The oscillation peaks during the daytime period, while another protein molecule, CCA1, is inhibited in a complementary fashion. This clock driven by sugar and interactions with protein molecules allow the plant to know the right time to photosynthesize, or otherwise the plant would try to photosynthesize at night, when doing so is completely unnecessary. All the genes involved in regulating their time schedules are used to maximize their development. The research expressed in photosynthesis entertainment of Arbedias thaliana circadian clocks provides insight into methods to promote plant growth in greenhouses where light conditions and plant circadian rhythms must be controlled. Farmers can utilize these processes, especially to maximize seedling growth and development, which can lead to increased crop yield. People will in turn have more access to food at a faster rate as a result of research on plant circadian rhythm. Crop shortages could also be a thing of the past.